Good morning, students. In the last class, in this chapter, we have learned some of the important experiments such as Griffith's transforming principle, and further the transforming principle of Griffith, how it is extended to prove that which is the transforming principle of Griffith's, what he has reported in the bacterial transformation. And along with that, we have also learned the one more experiment which has strongly helped the scientific world to prove that DNA is the only genetic material. So we just all recalled here whatever we have learned in the last class. So along with that, once we knew here that DNA is the only genetic material, why we have to consider as the DNA is the only genetic material, even though there is some viruses are having genetic material, RNA as their genetic material, why we have to strongly believe that DNA is the only genetic material. So the reason for that, DNA is going to satisfy certain criteria. So they are called properties of genetic material. Now let us see, once again recall what are the properties of a genetic material. Okay, so one by one, a genetic material, either it may be a DNA or RNA, so it has to satisfy all these criteria. Okay, so first criteria is the genetic material must be able to generate its replica, means it has to undergo replication, it has to multiply itself, it has to produce the exact copy of its own. See, if it doesn't have, if the genetic material, if it doesn't have this property, then you can imagine there would not be any uniqueness in the species, where we know that every species have a constant number of chromosomes. That is because of this, the replication property, you can say. Then coming to the next property, that is chemically, the genetic material must be chemically as well as structurally should be very much stable. So that we can just remember here the Griffith's transforming principle, where even though he killed the S bacteria, he could only destroy the bacterial cells, but the DNA did not be destroyed there. Okay, so it was only the chemically and structurally stability which is going to be proved from that experiment, that as the DNA, even the heat killed bacteria, yes, bacteria were heat killed, DNA was retained in the content, and then only it could transform the R bacteria into S bacteria. So that itself is sufficient us to understand, yes, the genetic material, what we consider strongly as the DNA, that is having this property, that is chemically and structurally it is stable. Okay, then genetic material should also have this scope for the mutation that is required for the evolution. That means genetic material should provide a limited scope for the mutation to take place, as we all know that mutations are going to induce certain variations among the offsprings. These variations are very essential for the ever changing environment to get the individuals adapt to their environment, surrounding environment successfully. So the third property is also very important. Then the fourth and the last one property, that is the genetic material must be able to express itself in the form of Mendelian pattern or Mendelian characters. As you all know that Gregor John Mendel, a father of genetics, he could gave certain the strong principles in the genetics. According to him, the monohybrid cross or the dihybrid cross, whatever the experiment he conducted, he mentioned there each genetic material here, it is nothing but it acts as in the form of genes, and these genes are going to control certain important characters, and these characters, if you take the two characters at a time, should express in 3:1 ratio. If you take more than two characters, that means the four pairs of contrast characters, that should express in 9:3:3:1. So this is what is the Mendelian principle or Mendelian pattern. So genetic material should satisfy this principle also. Okay, so these are the properties a genetic material should satisfy. Okay, so students, please read and prepare well, because in the 2019 this question is asked for 3 marks. Explain or describe or list the different properties of a genetic material. So be prepared for this. Next shall we move on to the next slide, that is, we have in the nature both DNA as well as RNA as the genetic material. Now, as we have both DNA and RNA as their genetic material here, then which one is the best genetic material? As we already know that DNA is the most and abundant genetic material, because almost all the living organisms contain DNA as the only genetic material. But even though that, we could also see there are certain viruses which contain RNA as their genetic material. Then among these two, which one stands better? If you take, okay, so we consider or we believe strongly that DNA is only the better genetic material. Why it is better genetic material? Because it's going to satisfy all the criteria just now we have discussed. So it's going to satisfy all the criteria a genetic material should possess. So what are that? The one is DNA is structurally as well as chemically, it is very much stable when compared to the RNA. Why it is structurally and chemically more stable? Because the DNA is existing in double-stranded nature. It is existing in double-stranded nature, hence it is chemically as well as structurally stable. So whereas RNA is existing in single-stranded nature. As RNA is existing in single-stranded nature and its two hydroxyl group, second, because it contains the pentose sugar, ribose sugar, where at the second carbon atom there is the freely exposed hydroxyl group, which makes the RNA most reactive and liable, means very sensitive, and it can get easily digested by the exonuclear activity. So as compared to RNA, DNA is not having any such what you can say sensitivity, so it is more strong and more stable because it is double-stranded in nature. Okay, and in addition to that, the presence of the nitrogen bases, that is ATGC, are the nitrogen bases in DNA, whereas ATUC are the nitrogen bases in RNA. So thymine is also going to give some additional stability to the DNA when compared to uracil in case of RNA. Okay, so as because of this strong structure stability, DNA is going to show the slow rate of mutation. Okay, whereas RNA, I said it is RNA, it is single-stranded, highly reactive, and it is unstable, so that is going to show fast rate of mutation. So because of this reason, every year during this time we are going to experience new varieties of viruses, as we are going to experience the COVID-19. Last year we are experienced Nipah, and before that Ebola, and before that H1N1, SARS. They are all varieties of RNA viruses. As RNA is highly reactive and undergoing fast rate of mutation, we could see every year the new type of viruses. So that is the drawback of RNA, not acting as a better genetic material. Now again, when you compare to the other part of DNA, so DNA being double-stranded and is chemically and structurally it is more stable, it is better to storage of genetic information. It is better for the storage of genetic material. For that purpose, RNA would never be better for the storage of the genetic information. RNA is better just to transfer the genetic information from nucleus, means from DNA to RNA in the form of messenger RNA. Okay, so because of this reason, we consider and we strongly believe that DNA is only the most suitable and better genetic material when compared to RNA. Okay, now we shall see some evolutionary history okay, about the genetic material. So evolutionary history says that RNA was the first genetic material. As you are going to study in the next chapter, that is evolution, that the first life which was originated on the earth, that was in the non-cellular form. That was in the non-cellular form, means that might be in the form of RNA or DNA or something else it is, okay. So that some of the evolutionary theories says that from the inorganic elements in the atmosphere there are certain changes took place, so that there is a recombination or reaction between the inorganic elements and the atmospheric changes made to form the first non-cellular form of life, that is RNA, okay. And this RNA, after exposing to the millions of years where through which the lot of evolution took place, a lot of environmental changes took place. So during that long period of time, the RNA has subjected for various type of evolution. So slowly and gradually it has transformed or evolved into DNA. This is what the evolutionary history says. That means that according to evolution, RNA was the first genetic material. As the RNA subjected for the millions of years, the long course of time, the evolution, then gradually it has changed into DNA. So according to this, which is the first genetic material, RNA was the first genetic material. As it was not stable and was very reactive and fastly mutating, so it has gradually evolved into DNA. And as now the DNA is most stable chemically as well as structurally, hence it is not showing much changes in the offsprings or in the evolution, we can say. Up to here we have learned certain important topics about the DNA and RNA. Now we are going to move towards certain important processes which we have to learn. So one among that is DNA replication. So what is DNA replication? So let me tell you first meaning of replication. What is replication? Replication is nothing but duplication. So replication is nothing but the duplication, okay. So DNA replication is nothing but the duplication of DNA. DNA replication is nothing but the duplication of DNA, where it is the, if we can define, means we can define it as it is the process of duplication of DNA, where the DNA molecule produces the exact copy of its own type. Okay, so DNA molecule produces the exact copy of its own type. So it's nothing but obtaining the one more Xerox copy from the original one. Okay, so DNA replication is nothing but obtaining the multiple copies of the original DNA, okay, either naturally or artificially. If it is artificially obtained, we call it as a polymerase chain reaction. Okay. If it is obtained naturally, it is nothing but DNA replication. Okay, so here it is the natural process of DNA multiplication or duplication, where the DNA produces exact copy of its own type. We can also tell this the DNA replication in other simple words as DNA synthesis or DNA polymerization. As we all know that DNA is made up of two polynucleotide strands. Okay, so each polynucleotide strand is nothing but the polymer of deoxyribonucleotides, right? So this process of duplication or the DNA replication is nothing but the DNA polymerization. Okay, I hope you understood the definition of the DNA replication here. In simple words, it is nothing but DNA polymerization. Okay, now who first gave the, actually here the many more models have been proposed to explain the mechanism of DNA replication, and such models include, one is the conservative model, other one is dispersive model, and one more model is semi-conservative model. Okay, so there are three models. You need to remember this as it is not mentioned in the NCERT textbook, but you need to know here for the point of your NEET examination. So for the mechanism of, to explain the mechanism of DNA replication, how many models have been proposed? Totally three models have been proposed. And what are that? One is conservative model, other one is dispersive model, and the one more is the semi-conservative model. Okay, so conservative model and dispersive model as could not give the proper, the convincing idea about the DNA replication were not accepted. Okay, as here semi-conservative model was the most accepted model proposed by Watson and Crick. Okay, and that is the one which is accepted most and was most convincing model for understanding the mechanism of DNA replication. How could they able to construct such a good and model for the DNA replication? Because they are the one who proposed the three-dimensional structure of the DNA, that is Watson and Crick DNA, what we call. Okay, so that DNA structure, as they had already the sufficient idea about the DNA, so they imagine that as the DNA is double-stranded, helically coiled structure, and based on their complementary base pair mechanism and all, they could propose this is the one model, that is semi-conservative model. So we shall see later the semi-conservative model. Okay, so before that we shall see that why these two models being not accepted and why only the semi-conservative model is being accepted. Okay, so here the important note about the conservative and dispersive model. So what, according to the conservative model, what it says that DNA replication, whenever it takes place, the entire parental copy, after each and every replication or round of replication, the entire one of the old parental or older copy is going to be retained, and another molecule of DNA will be newly formed. So that was not much convincing for the people. So that is why it was not accepted. And according to dispersive model here, dispersive model, as the name indicates here, it says that during the DNA replication, where the parental DNA is cut into many pieces, and alternatively around each and every pieces of the parental DNA, a new or the complementary copy of the DNA will get synthesized, and that is how the daughter DNA molecules will be produced. That means according to dispersive model, where the old or the parental DNA and the newly formed DNA are dispersed randomly. Okay, so as this was also not much convincing, so this was also ignored. Okay, then last come to the semi-conservative model, that is the two where this semi-conservative model, as I mentioned, was proposed by the Watson and Crick. Okay, so according to them, always the DNA replication undergoes by semi-conservative method. Semi means half or 50% of the parental DNA will be conserved in the newly formed daughter DNA. So according to him, from every one DNA molecule, the two daughter DNA molecules are going to be produced at every round of replication, and in each one of the daughter DNA molecule, one of the parental, that means 50% of the parental DNA will be conserved. So hence it is known as semi-conservative model of DNA replication. Now we shall see the diagrammatic representation. So with that you can understand better. So this is the one conservative type of model of DNA replication. As it says that this is the parental DNA and these two are the daughter DNA molecules. So here parental DNA is going to express as it is in the newly formed daughter DNA. So what you can say, model was not much convincing. And similarly dispersive model, here the parental DNA is going to be broken into pieces, and that every pieces will get synthesized alternatively with the new DNA molecules, hence it was also not much, you can say, convincing, so that is also ignored. And finally, which one was the most accepted model? That is the one semi-conservative DNA replication model, which was proposed by Watson and Crick. According to them, each DNA molecule which is double stranded and which is going to be separated into two single stranded DNA molecule, each one of that act as a template. On each one of the template DNA, or you can say parental DNA, a new strand is going to be synthesized, okay, automatically. So as one of the parental DNA strand is conserved in the newly formed daughter DNA, it is considered as a semi-conservative. Semi means half. So 50%, one of the strand of the parental DNA will be conserved in the newly formed daughter DNA. Hence it is considered as a semi-conservative method of DNA replication. Now, students, we should learn that why replication has to take place, what happens if the replication doesn't take place, and when the replication is going to take place and where the replication is going to take place is very important to understand here, okay. So as we all know that our body is made up of trillions of cells, okay, but all the cells have got the same amount of DNA. Okay, all the cells, you have to remember here, all in our body cells, the amount of DNA is same. How is it possible to retain all the cells with the same amount of DNA? That is because of replication. So this replication has to take place. Otherwise, what happens? It leads to certain complications. Okay, so that means, so some cells will have the half amount of DNA, some cells may have double the amount of DNA. That leads to a lot of complications. So to avoid that, replication has to take place, and it is taking place, and it has to take place, okay, throughout our life, because in our body every day the new cells are getting added, okay. So how they are added here? Through repeated mitotic cell division. So whenever any cell, before it is getting into divided to form a daughter DNA, it passes through a period of interphase before it divides to form a two daughter cells. It passes through a phase called, a preparatory phase of a cell cycle called an interphase. As you all have studied in the last year, that the preparatory phase, that interphase, the cell divisional wise is only a preparatory phase and a resting phase. As far as the metabolism is concerned here, it is a highly active phase, where in the G1 phase the cell is going to prepare itself for undergoing replication and further other things to take place. Then in the S phase, so S stands for synthetic phase. So what synthesis is taking place here? DNA synthesis. It is nothing but DNA replication is taking place in the S phase. So where the DNA replication is taking, when the DNA replication is taking place, replication in an eukaryotic cell, it is taking place in the S phase, okay. After the DNA replication, see, you can just change the, notice the changes here. Each chromosome represents one DNA. So after the DNA replication, see, each chromosome has got one more identical copy of its own type. That means exactly the DNA, whatever which is present in the parental cell, is doubled here, okay, and the same amount of DNA is going to be retained in all the daughter cells as well as in the parental cells because of the cell division. That means the replication and cell division both are very, very important in the life of an organism, should go hand in hand. So if the replication takes place, cell division does not take place, then again it leads to another type of complication, that is called polyploidy, where the chromosome number in a cell increases, that is triploid or tetraploid. So that condition again leads to certain complications. So to avoid that, it is very important that replication should take place. Then when it occurs in the S phase. Then where it occurs is also very important for the point of your competitive exams and all. You need to know, as far as the eukaryotic cells it is concerned, the DNA, as it is associated with histone proteins and held in a chromosomes present in the nucleus, so that that DNA replication occurs in the nucleus of the eukaryotic cell. Whereas if you consider a prokaryotic cell, in the prokaryotic cell, as they have a nucleoid, that means they do not have a well-defined nucleus where their genetic material, which is freely scattered in the cell cytoplasm, there itself, means in the cell cytoplasm itself, the DNA replication is taking place. Okay, now we shall move on to the next the slide, where we are going to focus on the semi-conservative method of DNA replication. So before to learn about the mechanism, exact mechanism of the DNA replication, this is one of the experimental proof which was proved that DNA replication always occurs by semi-conservative method only. Okay, so this experiment was being performed by two of the scientists. They are Matthew Meselson and Franklin Stahls. They performed this experiment in the year 1958 in order to prove that DNA replication always occurs by semi-conservative method. So how did they perform and what they took for their experiment? Here they took Escherichia coli bacteria. Okay, they took the Escherichia coli bacteria and cultured first in a nutrient medium, and that was containing ammonium chloride, where the nitrogen was made to have N15. Okay, nitrogen was made to have N15 in that media. N15 is an heavy isotope of nitrogen. Okay, then after some time, means after 1 hour or 2 hours, they transferred all the bacteria, E. coli bacteria, after grown in the ammonium chloride, where the N15 was present over there. Okay, transferred to the E. coli bacteria into the medium containing N14, and that means the medium containing ammonium chloride, where the nitrogen is of normal, that is N14. Okay, now so this they have grown at different time intervals in order to show that what is the percentage of the parental DNA is going to be retained at every generation of the bacterial progeny. Okay, so for that I have here the diagram which is drawn on the board. With the help of that you can learn better. Okay, listen students, here this is the diagram which for the Meselson and Stahls experiment I have little bit modified it. So this is as same as it is given in the NCERT textbook, and to make you understand better I have little bit modified it. So let us study now. So what is that experiment says? See, Meselson and Stahl, they have performed an experiment to prove that DNA replication is semi-conservative. So what they did, they took E. coli bacteria, they took E. coli means Escherichia coli bacteria and cultured in the nutrient medium, that is containing ammonium chloride, where the nitrogen is of N15. Okay, they cultured E. coli bacteria in the nutrient medium, and that is containing ammonium chloride, in which the nitrogen is made to have N15. So N15 is an heavy isotope of nitrogen, where like this in this medium the bacteria were grown for many generations. Bacteria were grown here for many generations. Okay, and then finally what happened after the many generation, this was the only source for the nitrogen for the growing bacteria, and their entire DNA, the nitrogen bases were incorporated in the DNA made up of that N15. So that the entire DNA of this group of bacteria that incorporated with N15 in their nitrogen bases. Okay, and this group, some of these bacteria are taken, and the DNA of those bacteria was isolated and means extracted. Okay, then centrifuged by using the cesium chloride solution. Okay, by using the technique, the density gradient centrifugation technique, okay, in the centrifugation tube. So this DNA containing, this group of bacterial DNA containing heavy isotope of nitrogen, the density being heavier, this group bacterial DNA was settled almost at the bottom of the test tube, okay. Then some of these bacteria, means this group of bacteria are transferred into another nutrient medium where the normal ammonium chloride was taken there. That means these bacteria, some of these bacteria were transferred, okay, were transferred into the nutrient medium where the ammonium chloride which was taken here was normal ammonium chloride, where it had N14 instead of N15 here it is taken, here N14 it has normal nitrogen, okay, and which were allowed to grow for 20 minutes, which were allowed to grow for 20 minutes, and after the 20 minutes, and this group, some of the bacteria of this group were taken and DNA of those bacteria was extracted, again centrifuged in the cesium chloride solution by density gradient centrifugation technique, and in this DNA being hybrid or intermediate, okay, so this was settled almost in the middle of the test tube. So it indicates that it is neither heavier nor lighter. So it is being intermediate or hybrid. It is settled in the middle of the test tube. Okay. Then these bacteria were allowed to grow in the same medium for another 20 minutes, means 40 minutes. After 40 minutes, again some of these bacteria were taken, okay, and the DNA of those bacteria were extracted, okay. Then once again those bacterial DNA was centrifuged again by using cesium chloride by density gradient centrifugation technique, and after that what did they found, that the one part of DNA which was settled almost at the top of the test tube and another part of the DNA which was settled almost in the middle, that is just like the first generation bacterial DNA. So that indicates that the three different group of bacteria had got three different density of bacterial DNA. That means here the bacteria, so one example I have taken here. So when the bacteria are grown in a ammonium chloride where the nitrogen was of N15, for many generation those bacteria will take entire their DNA, that N15. Okay, entire DNA will be incorporated with N15. So N15 is a heavy isotope of nitrogen. So hence the DNA being heavier settled at the bottom. Then after 20 minutes, if the same bacteria is going to divide to form a two-daughter bacteria, so before the cell division, this bacteria is going to double its genetic material, means duplicates its genetic material. So if the two strands of this bacteria are having N15 in their DNA, the two strands will get separated into two daughter DNA as templates, that is N15 will pass on to the one bacteria, another N15 strand will pass on to the another bacteria. So immediately on each of the template DNA in both the bacteria, you can see the normal DNA which is going to be formed here, incorporated with normal N14 nitrogen in their nitrogenous base pairs. And similarly, after one more generation, means after 40 minutes, if these two bacteria were allowed to grow for 40 minutes, what happens? Two bacteria will again undergo binary fission to form a 4 bacteria. So these 4 strands of the two bacteria will get separated into 4 single-stranded bacterial DNA, and on each one of the strand in each daughter bacterial cell, the new copy of normal DNA will get synthesized. Okay, so hence the two bacteria will receive the hybrid DNA here, and the rest of the 2 bacteria will receive only the lighter DNA. And here after 20 minutes you can see that the completely DNA is hybrid. So being with this variation in the density means depending on the N15 and N14, the density gradient centrifugation technique is going to indicate that the DNA being heavier, here it is settled at the bottom. Here the DNA being intermediate or hybrid is going to settle in the middle. Here the DNA being is of 2 types. So one is hybrid type, another one is lighter, means completely N14 type is going to settle at the top. So this indicates that at every 1 generation the parental DNA, 50% of that is going to be transferred to its progeny. So here the 100% heavier DNA. So this 100% heavier DNA to the next generation is going to be transferred, means it's 50% of the DNA is going to be transferred to the next generation. So here 100% it is hybrid DNA, but here 50% of N15 heavier DNA is incorporated, and remaining 50% will be lighter DNA. Hence it is hybrid, but 100% hybrid it is. Then similarly what happens here in the next generation, here the 50% hybrid DNA and 50% what you can say lighter DNA. So once again the 50% of hybrid DNA is going to be retained 50% of that. So 100% of hybrid DNA is going to reduce us in the next generation into its 50%. Okay, then rest of the 50% will be of lighter DNA. So this is how what it indicates. After 1 generation the DNA is going to be reduces. So it's 50%, means heavier DNA or hybrid DNA reduces to its 50%. That means one of the parental DNA strand would be passed on to the next generation as it is. On that the new copy of the DNA is going to be synthesized. Okay, so this is what the entire experiment is going to say about the DNA replication is semi-conservative. Okay, so this you have to learn, for the point of your examination, very much important it is, as it is asked in the last year, in the year 2019 March question paper for 5 marks. So you need to learn it very comfortably, and you have to extend. So in your textbook they have given that what happens if it is extended for further 80 minutes, that means for 3rd and 4th generation, what is the percentage of the heavier DNA, lighter DNA, as well as the complete N15 DNA, hybrid DNA. So that please work out and tell me the answer.